All right, guys, tonight's special guest speaker, Dr. Michael Grace, is a professor emeritus at Florida Institute of Technology. Previously, he served as professor of biological sciences and senior associate dean of Florida Tech's College of Science. Dr. Grace is an expert in the field of marine vision. He holds a master's degree and a PhD from the Emory University School of Medicine. He spent his career researching the cellular and molecular mechanisms of vision and the effects of light on different organisms. Dr. Grace has studied visual and sensory systems in a wide variety of really fascinating animals, including pit viper snakes, pythons, marine fish, sea turtles, even whales. His work has been featured in National Geographic magazine, on the Discovery Channel, the Learning Channel, National Geo Wild, CNN, BBC, ABC, as well as a bunch of local news networks. Tonight, Michael will discuss the unique aspects of vision in some of our most beloved marine organisms and how insights into marine vision may tell us some things about human visual disorders. It is with great, great pleasure that I welcome my good friend Michael Grace to the stage.
A second. There we go. All right. Well, thank you very much. Thanks for that warm welcome. It's really great to be here. I'm really impressed. What a great crowd. So I hope that I don't disappoint. It's interesting how I came to marine biology. I actually grew up landlocked. Always loved the water, always loved being on the water. But I grew up far from the ocean, would come down every now and then. As a matter of fact, I'll tell you, I think I was in about the third grade, maybe the summer after third grade. My parents brought us down, and that whole year I dreamed about it because we were going to Sanibel Island. And I had heard about it, the shelling capital of the world, and I was so excited. And we came down, we actually stopped in Tampa, and we're going to spend a few days there. And I was excited about that, but it turned out that the red tide had been moving north up the coast. And my parents were just mortified. It really smelled, you know. And I was so sad, though, that we had to leave and go on south to Sanibel because I was in heaven. I was a little kid, and all these amazing creatures, unfortunately, dead on the beach, but that meant I got to see them. And all I wanted to do was load up the trunk of the car, take all of these things home. They nixed that pretty quickly. However, when we got down to Sanibel, red tide was moving north, so it turned out that, the beaches were still littered with all sorts of interesting things, but they were a little drier, a little less smelly, and they actually let me bring a few things home, so I was thrilled. So every now and then is a good story. It helped motivate me, I suppose. Well, this thing's kind of going in and out. Hopefully it'll hang in there. I studied neuroscience. I studied cell biology. I started out at Georgia Tech and then went to Emory and then went to the University of Kansas Medical Center. Loved animals my whole life, but I had this notion that you needed to do something that was more practical, something that people actually cared about other than me, playing with snakes and things, and might not earn me a living. So, and it really was me, not my parents. I had, in fact, people around me were telling me, do what you really want to do and you'll be successful. But I didn't do that. I went routes that I thought I should go. Eventually, though, I started taking the things that I had learned through that training and applying them to questions that I was really interested in. And honestly, the big change came when I finally got a grant from the Department of Defense to study infrared vision in pit vipers and pythons. That just, it gave me the freedom, I suppose, to go and explore something that I'd always loved, and it also gave me, some cachet, I suppose, to be able to build a career in the direction that I really wanted to go. Again, taking advantage of the things that I had learned in those other environments. And then, instance, I ran across an ad for a position at the Florida Institute of Technology, and it looked like it was written for me. I had never heard of Florida Tech. I had never heard of Melbourne, Florida, but they were willing to fly me down in February for an interview to the beach in Florida, and I was ready to get out of the snow and the cold, so I said yes, and they hooked me. I loved it. So I stayed there for 20 years. I've really, really enjoyed it, and being there, I couldn't help it. I got drug into the world of marine biology. So it was kind of nice, though. I got back to those days on Sanibel in Tampa. So what I want to do tonight is tell you a story about, really two stories, at the interface of marine biology and vision. And as a graduate student, this is where I got started in vision. I've always been an avid photographer. I've always loved the way animals interact with their natural environments and how things like light affect them. So it was sort of a natural thing to go into the science of vision. And again, moving to Florida, began to apply that to the marine environment. So I'm going to tell you about some extremes of vision, and I'll tell you why they're extremes. I mean extreme relative to us. And so we'll talk a little bit about our own visual system and how it relates to these animals. So I think this is a pretty smart audience, it seems to me. So this is a I could give in multiple places. Hopefully, if you have any questions, we'll certainly delve into it a little deeper if you need to. But there's a lot of data. But I'm going to hopefully explain it so that it makes good sense. I promise. So again, extremes of vision. Extremes in two senses, mammals and fish. Extreme in the sense of size, but also extreme in the way vision in these animals work. Will be fun. So some of you know, if we have vision problems, we may know more than some others, a little bit about how the visual system works. So at the back of your eye is this wispy, thin little piece of tissue known as the retina. We've all heard about retinas. We know about retinal detachment, things like that. But you would be amazed if you saw the retina. It just looks like a thin, wet piece of tissue paper, thinner than that even. It's almost nothing. This is a gross exaggeration of the thickness of the retina at the back of the eye. Tiny, tiny little piece of tissue. But within that very tiny piece of tissue are millions and millions of cells that capture light and process light. And a lot of what we do in terms of seeing the world around us is all done in the eye before that information ever gets to the massive brain for processing. So it really is amazing. This is the architecture of the retina is very precisely laid out. If you're an engineer, you would love the way this thing is built. And again, millions and millions of cells all talking to each other to do things like detect whether there's light or not, detect the spectral qualities of the light, the color of the light, detect the intensity of the light, detect edges so that we can see light versus shadow, and even detect shapes of things, recognize things like, at least rudimentary things like shapes, even faces, and even motion. All of this is done in the retina before this ever gets out to the brain for further processing to really help refine what the retina has already picked up. So it's an amazing piece of tissue. Whoops. But I'm going to focus not on the whole complexity of the retina. I don't know what I'm doing. But rather a couple of different cells within the retina. So I wanted to introduce you to one of my heroes. Santiago Ramon y Cajal was a scientist, an anatomist back in the 1800s. He's my hero for a number of reasons. First of all, he developed some amazing techniques that we still use today to stain tissues so that we can see the organization of cells. And his particular technique, known as the Golgi method or silver staining, generally Golgi is slightly different. But it uses some chemical properties of a colloidal silica solution to fill cells, and nervous cells will actually take up this material and it will fill the entire cell all the way out to the finest branches of that cell, so you can really see what a really looks like. And if you know what that cell looks like, then you can begin to understand the connections that it makes with other cells. And his work, his development of this technique revolutionized neuroscience way back then and still does today. So the other reason he's my hero, I should tell you, he was a real troublesome child. I've read a biography of him, and my favorite story, I grew up in the country on a farm, and there's a story that when he was a young child, he found this piece of pipe, a very heavy pipe. I don't remember what it was made of, but he got this great idea, and his farm, his place was up on a hill overlooking the neighboring farm, and there was a big stone fence down there with a giant wooden gate. He set this pipe up there and found some gunpowder and stuffed it full of gunpowder, and then he found whatever he could, nails, rocks, pieces of wood, and stuffed all that in there. And he just thought this would be a great adventure. And he lit this thing, and as it's about to go off, he notices the farmer coming over the hill. And boom, this thing sets off, and the farmer sees his giant wooden gate just explode into a million splinters. And that was the beginning of one of the times that Cahal ended up in essentially a reform school back in the day. So if I got in enough trouble when I was a kid, if I had read his biography, I would have been in a lot more trouble. Anyway, he helps us understand the retina way back in 1892. And he and a lot of other people, put these quotes up here, they really, lots of people and I, we sort of take vision for granted, but it is so fundamentally important to us as a species. We are very visually guided species, and many more species on this planet are as well. So I particularly like, I don't know if you know Khalil Gibran, he's a philosopher, a poet, but he says the eye of a human being is a microscope which makes the world seem bigger than it really is. Lots of people have expounded on vision as so fundamentally important to everything that we do and think about. So this diagram, if you actually compared it to Cahal's diagram, looks pretty much the same, and this was drawn much more recently. And again, lots of different cell types, lots of different names for all these cell types, not just the cells, but also the connections that they make with each other. This is called a plexiform layer. It's just where all the little wires meet and communicate. But anyway, we're going to simplify this. What we're going to talk about tonight is only this part up here at the very top. Well, next to the light comes in from the bottom of the screen, and it actually passes through, first of all, it goes through your cornea, then it goes through the lens, and it goes through the big open space in the eye, vitreal chamber, and then it hits the retina. And those photons of light go through all of these layers of cells. It's actually very transparent despite all of this, and eventually it hits these photoreceptor cells, or light detectors at the back of the eye. Beyond these cells, these little guys here, they're called the retinal pigmented epithelium. This is what makes the back of your eye dark, and it is melanin. It's the same pigment that we have in our skin and our hair. Albinos lack melanin, and that's why the eyes look pink. But what that melanin does is it absorbs those photons of light that actually make it all the way through the retina so that that light's not bouncing around and degrading the image that's formed. The way I like to think of this is all of these photoreceptors are light-receiving pixels, in a sense. So all of this light is flooding into your eye. An image is inverted through the lens and projected onto the back of the eye. And so that dark space falls on this one particular part of my retina, and then a light space on the adjacent part, and another dark spot on an adjacent part. You literally have a mapping of the visual world on the back of the eye. And that map is generated by these photoreceptors. Oh, there I go again. And all of that information is preserved in a spatial manner as it moves through these different cell types and eventually out to the brain. You literally have a map of this visual world in your brain right now, a constantly changing map. But it's literally physically mapped on the retina and in your brain. Anyway, back to the point here is if we look at these photoreceptor cells, this is a diagram of what these photoreceptor cells look like. And again, same orientation, light is coming up from the bottom of the screen, it's passing through the cell body, this is where the nucleus of the cell is, and it's captured out here at this far end. This is called the outer segment, it's the receiving end of the cell. There are molecules in here, collectively, commonly called rhodopsin, that actually absorb the photons of light. And when light hits that rhodopsin, it sets off a chain of chemical reactions. Oh my gosh. Whoops, oh no. Okay, maybe we can get reset. I know part of what I'm doing. I'm pointing it back here. I should be pointing it that way. All right. So the business end of the cell is way out here at the end. A series of chemical reactions takes place in response to those photons coming in, and they either excite the cell or cause the cell to stop being excited, and either way, that's information. So that's generally what a photoreceptor cell looks like. They all sort of look the same, but if you notice out here, there are some red and green and light blue ones, and then there are these more elongated dark blue ones. Those are both photoreceptors, but there are two different types in us and in most animals. There are so-called rod cells, and there are cone cells. So the cone-shaped ones, they are literally, that outer part that collects the light is literally shaped like a cone, and the rod cells are shaped more like a rod. So someone was very creative when they named these. So, but they look different, but they're functionally different. Rods give you dim light vision. This light is still bright enough that you're activating your cones and probably some rods as well. But if you go out on a moonlit night, only your rods are activated. There's not enough light to activate the cones. They require a lot more. The rods are very sensitive, so they can detect that dim light. Cones aren't activated. In the daylight, the cones are activated. There's enough light to activate them, and there's so much light you actually swamp the rods. They just can't handle it, so they turn off. So notice, though, if you go out on a moonlit night, you don't see color. Sometimes we think we see color. If there's, two apples sitting there, one's, golden delicious, and one is, what's a red one? Red delicious. There you go. I'm just seeing if you're on your make sure you're awake. Macintosh. So you see those two, and you and at least I do. I think, oh, that's the red one, and that's the greenish or yellowish one, but you really can't see it. That's your memory knowing what it is. You can't see color in dim light because there's only one type of rod cell. There's many, millions of them in your retina, but they all work the same. It's either light on or light off. That's all they do. In highlight, though, the cones give you color. And the reason is there are 3 different types of cones, and they have different spectral sensitivities. They're activated by different parts of the visual spectrum. So, again, this is in us, in people. If you look at a graphic arrangement of how these different photoreceptors are activated by the visual spectrum, the dotted line is your rod. They are activated best by green light and to some degree by blue and yellow, maybe a little bit into sort of an orange. That's just the way they operate. But there's only one type, so you don't see color. They're either turned on or turned off. Actually, deep red, you can't see at night because your rods just aren't sensitive to that light. But the cones, again, 3 different types of cones. If the light is bright enough, these different types of cones can be activated. We have one that we typically call a blue cone, one that we call a green cone, and one that we call a red cone. Cones are activated primarily by light in the blue part of the spectrum, the green into the yellow and orange, and the red cone is activated by red and down through the yellow and green as well, but it's the one that's activated best by red, so we call it a red cone. So how do you get all this color out? Your brain basically just computes how much information is coming in from each one of these channels. How much blue, how much red, how much green. And simply based upon that, and these millions and millions of different cones scattered over the entire surface of your retina, you can detect this vast array of colors that we see. Is anybody here colorblind? Yeah, there's at least one. Does anybody know how color blindness works? Yeah, so most colorblind people are simply missing one of these 3. Colorblind people are missing 2, And very rarely there's a person who's missing all three. I've never heard of someone, but it can happen. But most colorblind people are missing one of these three classes. Still have color vision because you've got more than one type, and your brain can still interpret color based upon that. But with only two, your ability to discriminate color is pretty severely diminished relative to normally sighted people. As a matter of fact, among mammals on Earth are what we call dichromats, two color channels. They're like colorblind people. They have only two. Great apes and generally old world monkeys have three like us. New world monkeys, some, alone. New world monkeys we generally think of as only having two. Great apes people have three like us. There's a caveat to the New World monkeys. They're really weird. But anyway, so this is us. This is humans. Normally sighted people, three different types of cones. Your brain interprets all this color. Amazing array of color, just by sampling how each one of these types is activated. With this as a background, what I want to tell you about is how submarine fish see and how whales see, especially with respect to color. This is just a little diagram of the protein that is the light detector. It's called opsin, collectively called rhodopsin, when it's the protein plus a molecule of vitamin A. People always say, you got to eat lots of vitamin A. Without vitamin A, you don't see, because it's actually the vitamin A that detects the light, and that vitamin A changes its shape in response to light, and that causes the protein to change shape, which activates the whole cell. Anyway, it'll be important. Yes? Say it again? So the question, what do these letters represent? R represents rod. We'll often call this a blue cone, a green cone, a red cone. The other way we call them is a short wavelength cone, a middle or medium wavelength cone, and a long wavelength cone. So long wavelengths, short wavelengths. Good question. Good eyes. Good eyes, I like that. All right. I love animals. I love animals and I love pretty things. And to me, this is a very pretty animal. The reason I put this in here, I've actually done a lot of work on reptiles. I love reptiles, but this talk has nothing to do with reptiles, but I just wanted to point out what a gorgeous animal that is and how important vision is to us, including this animal as well. But what would it be like if we lost our color vision altogether? It would be a much less colorful world, to say the least. Or if we lost our vision altogether. So, fortunately, this is what our world looks like. So, I want to tell you about a couple of animals that live in a relatively blue world and how they deal with life in that blue world. So you probably are all very familiar with the tarpon. You either or see them out there or have one swallow your arm up to your shoulder or who knows what. Tarpon are amazing animals. I actually, this is one of the great things about being in academia. A graduate student got me in love with tarpon. He loved tarpon. He came to my lab. He said, I want to work on tarpon. He took my class in sensory neurobiology. He said, I want to take the things that I learned in this class and figure out how tarpons see. And his work was the basis of a lot of follow-on work, that part of which is shown here. I've had the good fortune to work with some outstanding students who also love tarpon. They start out larval form known as a leptocephalus. They have a tiny little head, a long flattened body. This is actually a big leptocephalus. It's got some growth on it. And these very tiny eyes. In fact, you see that dark spot? That's the melanin. That's the pigment at the back of the eye, behind the photoreceptors. That's how we see their eyes and understand what we're working on. So, tarpon and their relatives begin life as this planktonic larval form out in the ocean. move inshore. They come in through the inlets and settle in the lagoon and places like this around the world, and they metamorphose. They change from that strange ribbon-like thing into something that looks more like what we think of as a regular fish. And the juveniles live in these nearshore waters, inshore waters. They have a very different life. They have different kinds of predators for different kinds of, and they obviously they look very different, but they really behave differently. They have a whole different lifestyle once they move inshore. And then they ultimately mature into an adult where they move back offshore. This is why we really got excited about tarpon. This is an animal that begins as this tiny, tiny little thing just floating around in the sea at the mercy of everything around it. They probably migrate vertically in the ocean as the days progress. But then they move into these inshore waters that are typically darker. They're often stained with tannins. The water actually has a reddish color. You may not notice it, but if we measure it with our instruments, it's much more reddish. And then, as they mature into adults, they go back out offshore into this clear blue water, much less turbid, shifted back down to the blue. We were wondering, how in the world can a fish survive? A visually oriented, at least as an adult, predator. How does it survive these massive changes in the visual world? So we wanted to figure that out and try to understand the development of vision in this animal. And as a byproduct, we actually studied the other members of the group to which tarpon belong. The tarpon, ladyfish, bonefish, and eels are all part of the same group of animals. Many kinds of eels, and they live different sort of lives depending on the species. The one we really focused on because it's so common here in the lagoon is worm eel. Very, very common. We could get them very easily. They live down in the sediment. They tend to feed at night. They're buried most of the time. But then you've got this tarpon that's a mega predator that feeds at or near the surface, daytime, nighttime. And then you've got other fish like bonefish, for example, that is typically a bottom feeder and very diurnal. So you got very different lifestyles within this group. So not only did we want to know how they change over the course of life, we wanted to the different species and try to understand the differences, how their differences in vision might relate to how they live. For all we knew, everybody is exactly the same. They never change as they grow up. There's no differences among species. But fortunately, it turned out to be more interesting than that. So that's what I want to tell you about here. So do they all see the same, all the different species? And does their vision stay the same, or does it change as they mature through all of these different life stages? All right? So, the larvae. These are some close-up shots of the heads of those little leptocephali. They kind of look like little monsters. Many of them have these long, forward-projecting teeth. We really don't know much about how they live out there. Some people say that they just drift around and they absorb things through their skin, their nutrients. Other people say that they just bite off hunks of this stuff called marine snow, this gelatinous material, but really nobody knows a lot about how they really live out there in the wild. But we were able to collect some of these as they came through the inlets into the lagoon. We'd stay out all night with nets hanging off of bridges and hope to find one or two or maybe three of these little larvae. Then you take them back to the lab and you spend days and days and days trying to figure out which type you have because they all kind of look the same. But eventually, with a book about this thick and some weary eyes and a couple of microscopes, you can figure out which ones are which. We looked at a wide variety of eels. We looked at tarpon, ladyfish, and bonefish. And the short of it is, when they start out life, everybody looks like all they have are rods, no cone cells. So the dim light photoreceptors, none of the bright light photoreceptors, and probably no color vision when they get started in life. But it turns out that over the course of life, things get really complicated. In the very earliest stages of the tarpon, the very earliest stages, they really look like they're all rod. It's hard to get them when they're brand spanking new because they're somewhere offshore and you just can't find them. We've actually spent weeks out on trawlers trying to pull larvae out of the ocean and had essentially no success at all. But by the time they move in to the lagoon, they're actually starting to metamorphose as they're coming in through the inlet. So you catch them early, you've got a fairly early stage, you catch them a little bit later and you can already tell they're starting to shorten up, they're already starting to change morphology. And what we see during that change stage is, first of all, I should have told you what you were looking at before. This is an actual photograph of that retina that I showed you before. This is the back of the eye, light would be coming in from here, and that dark stuff, that's the melanin. It's the dark pigment that's back there behind your photoreceptors, behind yours and behind the tarpons. All of the other cells that I mentioned are down here. All these little blue dots are the nuclei of the other cells. So this is the inside of the retina, that's the back of the retina. Light comes through, hits the photoreceptor, and that's the first stage of seeing. Down here, we use a technique that is so cool. If you vision, you like color vision, you love this because it's pretty. It's a technique known as immunofluorescence. In a nutshell, what we do is we take antibodies, the same kind of antibodies that are made in us to fight against disease, and we can tag those antibodies with fluorescent molecules. And the nice thing about those antibodies is a particular antibody that goes after a particular protein on a cold virus, that's all it does. Another antibody that goes after who knows what, some other little tiny segment of protein on something else, that's all it does. So we have ways now where we can actually make antibodies, tons of them, that are very, very specific for very specific things. What we do here is used antibodies that recognize nothing except the cone opsin protein or the rod opsin protein. Remember the protein, the opsin, that's the first molecule in your eye that actually detects light. So we're actually looking for cones, looking for rods by looking for the protein that's in them, by tagging them with this fluorescence. Throughout this talk, what you'll see, I'll show you a few more of these, the red is rods and the green is cones. Again, they don't look like this in nature. You take them in the lab, you make tiny little sections, and you put all these chemicals on there, and a few days later, you look at them on a fancy scope, and you see these pretty colors. But they tell you where the rods are, where the cones are. Very early in life, we see rods, lots of rods. And by the way, the reason we do this is because the shape alone doesn't necessarily tell you everything. Sometimes you can be fooled for various reasons, but this really tells the story. So if it's red, it's rod. If it's green, it's cone. So a little later, as these guys are just beginning this process of metamorphosis, transforming, all of a sudden you see green. They start adding cone cells to their retina. And this is a diagram. This is not real. We actually just painted this in the computer. So you can see the distribution over the surface of the eye. They tend to add cones back here in the back, sort of the forward-looking part of the eye. They still have rods there as well, but that's where the cones begin. And then they spread out over the whole eye. I'll give you a little aside here. Well, what I'll show you is over the course of the life of this tarpon, that eye goes from nearly microscopic to gigantic. It's about as big as a baseball, much bigger than a cow's eye. Much bigger, much bigger. So there's a dramatic change in this eye. How do you fill up that eye? You don't just make bigger and bigger cells. Cells don't work that way. They're tiny. You put a lot more cells in there. Millions, I don't know, maybe billions more cells to fill up that giant eye. So over the course of life, this animal is adding more and more and more of all of the different types of cells in the eye, including these photoreceptors. So you start out with rods, you begin adding some cones, and then, as we, there we go, oh, I'm skipping some here. So, as these larvae continue through that maturation process, once they're in the lagoon and they really are starting to look like a normal tarpon now, all of a sudden, you still see this wide distribution of rods, but lots more cones, and they're spreading out over the entire retina. The rest of this are some other microscopic techniques that we use to prove to the other scientists that these really are rods and really are cones. Nobody trusts you in the scientific world. You have to, no, I swear we're telling the truth. It's right. All right. Ma'am? Mm-hmm. It's a good question. The question is, to paraphrase, if I may, the way I think of it, but it's exactly the same thing. Are these changes happening because maybe of a genetic program, and that prepares them for what's about to come next, or maybe it allows them to go out into they need to seek out a different type of water? Or does it happen? Do these changes happen because of the environment they're in? We actually think it's kind of a combination of both, that there is sort of a genetic pre-programming, but also when they seem to make these shifts into a new habitat, a new light habitat, that tends to accelerate, we think, what's really happening here. So are they doing it so that they can change, or do they make these changes because they've changed environment? It's not entirely clear yet, but it seems like there might be a little bit of both going on. But it's a great question. So a little bit more about how this animal develops. This is, juvenile moving into, we see some more big changes. Again, the cones are spread all over the entire retina. Each one of these little sort of bits here, that's actually a single cone. But notice what's happening. When we first saw this, we were absolutely baffled. We thought we had damaged the tissue or our technique wasn't working. Those giant blobs, what are those things? It really threw us for a loop. But it turns out they are rods, but by this time of the animal's life, the rods are actually clustering into these big bundles. Each one of these individual things here is a single rod outer segment, the part that gets colored. So each one of these is a single rod, but they're all clustered together. We don't see that early on, but we see it later in life. And this is another diagram. So back of the eyes, light is coming in this way. The photoreceptors are from about here to about here. These big massive things are the bundles of rods. There are hundreds of rods in each one of these things. So big changes going on in this animal's retina. So here's another change. This is something we didn't see early on, but remember I told you that back here right behind the photoreceptors there's all this dark black melanin, and that absorbs any photons that make it all the way through. So they don't bounce around and activate receptors that are in other sites and degrade your image. So that melanin is really important, but by the time we get to the adult, take my word for it, there's less melanin back here. These little black dots, that's all the melanin back there. There's not as much as you see earlier on. But around these rod bundles, we see this other stuff, this sort of granular looking stuff. And it turns out something called guanosine. It's these crystals of guanosine. And what that is, there's this layer that develops in the eye of this fish right behind and around the photoreceptors that's quite the opposite of that dark melanin. These guanosine crystals are highly reflective. If you dissect one of these eyes, and by the way, I should tell you, we have permits for collecting larvae and a few juveniles. The adults, we go to legal tournaments, and if a fish dies on the line or if a shark, kills the fish, that's where we get our adult eyes from. So we don't go out and kill tarpon for this work. But anyway, highly reflective crystals, and you may know this from other animals, we call it eye shine. When the light hits that tarpon's eye in the nighttime, it just reflects back at you like a mirror. So the guanosine crystals are actually reflecting light. So why in the world would you start off with an eye that has this black pigment at the back to give you a better image, and by the time these guys get to be adult, they built these giant mirrors all around the light detecting cells. We think what that means is it gives them better dim light vision. Where you see eye shine is typically in animals that are nocturnal. Turns out tarpon are diurnal and nocturnal. Anyway, I'll add all of this up for you in just a second, but I want to tell you one other thing that happens in their eye, and this is cool. On the left side, we've got tarpon from the light, and on the right side, actually, this is tarpon. But we see the same thing in ladyfish. Tarpon and ladyfish are pretty much the same, bone eels very different. But anyway, in tarpon and the ladyfish, in the light, the rods are here, again, back of the eye here, beginning of the retina over here, light's coming in from the bottom, going to the top. These are those bundles of rods. The cones are down here. We can tell by the shape, and again, we've done the fluorescent stuff too. At night, the rods are way down here, not out here. They were here, they've moved into here. These little blips right here, those are the cone cells. They were way down here, and now they've moved out there. The cells in the eye move around every day. They switch positions. So in the nighttime, the rods come closer to the incoming light, and the cones get shoved out of the way. In the daytime, the cones pull in closer to the incoming light, and the rods get shoved out of the way. It is a really cool adaptation. We call it retinomotor movement. Something's moving around in the eye. And again, what this is an adaptation for an animal that can operate very effectively in both light and dark. Some other animals are known to do this. We don't do it. But a really cool adaptation in these tarpon. Let's see. So this is the last bit of data for the tarpon. But I want to tell you about a little tool that we use known as Microspectrophotometry, MSP. I alluded to it in the introduction. What we can do is put a retina, any piece of tissue really, in a solution that contains some enzymes. You ever use, Adolph's meat tenderizer? This has got papain in it. It's an enzyme that breaks down protein. We'll put it in some enzymes like that kind of breaks down the connections between the cells. You have to do it really carefully. You don't want to destroy the cell, but you want to break down the connections, and you kind of shake it. And what you'll see is those long, skinny outer parts of the photoreceptor, they'll just fall off. And you can actually see it with your eye. It'll be like the liquid sort of becomes cloudy, this sort of very faint, wispy white cloudiness. And then you take the rest tissue out, you suck up a little bit of that solution that has those bits of the light detectors in it, put it in another dish, and what you can do is put it in a special kind of microscope that shines a light through your slide that has those little bits on it and up to a mirror and to a detector. So you've got a microscope, you can actually see these little bits of the cone cell and the rod cell, and if you really have a good lens on it, you can tell the difference between them. You can see the shape and tell the difference. So looking through the microscope, I can actually find one of these tiny, tiny little things and aim this light directly through it, and that light comes back up here and goes to a detector. The light that comes in, we can tell the system what color light we want. And we basically march from blue to green to yellow to orange to red. We go through the whole spectrum. And what we do is simply say the little piece of a cell that we have sitting right here, what color does it absorb? Those red cones absorb red, and they let the other colors pass through. The green cones absorb green and let the other colors pass through, and so on. And basically by doing this, what we do is we get an image. There we go. We get these curves of spectral sensitivity. These should look like the curves you saw before. These are two different cone cells. They actually are kind of squished together in nature, but there's a cone that absorbs maximally in this part of the spectrum and another one in this part of the spectrum. So this is what you saw before. This is exactly what we collect with this technique. So this is people. This is us. There's a blue cone, a green cone, a red cone, and there's your rod. This is what a tarpon looks like. By the time it becomes an adult, we have a rod and 1, 2, 3, 4, 5 different kinds of cones. Normally sighted people have 3. These fish have 5 by the time they're adults. It's trust me, unheard of in the animal world. Most mammals, 2. Very few mammals, 3. Some animals, extraordinarily rare, have 4. It's unheard of to have 5. Tarpon are amazing. In fact, it's not just the number that they have, but notice where this one is. This is down in the ultraviolet. We can't even see this. They can see ultraviolet light that we can't even begin to see, right? And what does it mean to have 5? What it means to have 5, we think, sort of making a rough calculation from, say, most mammals and colorblind people to normally sighted people, you go from 2 to 3 to 5. We think that these guys can discriminate color probably 10,000 times better than we can. They are amazing at seeing color. All right, so let me just tell you in a nutshell where all this goes. I'll show you 2 extremes. The speckled worm eel, they start out life with rods, and they mature. They look like eels the whole time, and the retina looks the same the whole time. They have rods, period. Dim light cells. They live on the bottom of the lagoon. They feed at night. They don't need color vision. They don't see highlight. They never see it. They don't need color vision. They don't need a highlight detector. Tarpon, on the other hand, just like eels, they look alike when they're babies. The retinas look just alike as babies. As they mature, they start adding cones. They add one type and another type and then another type, millions of each of these. By the time they're adult, though, these tarpon have bundled rods, which we think makes them really good at night. They have 5 different types of cones, which makes them amazing in the daytime. And they also have that reflective tapetum, that eye shine, that makes them great at night. And they have retinomotor movements. These cells move around, making them great at day and night. So it really explains how they live, how they feed. Day, night, dim light, bright light, great color vision, fantastic predator. All right, I think the lights up and down tell me I'm waxing on. Should I tell you a little bit about whales? You sure? What? Are we good? Okay. Maybe my eyes are having trouble. All right. So the whale story is actually very short. It's short for a reason. I said extremes of vision. This is extreme. This is extreme. Now, you may have heard there are some interesting animals, some invertebrates that have even more color detectors than this. Maybe somebody, if you know about it, be a good topic for later. But in the vertebrate world, this is extreme color vision, no question whatsoever. So, short story on whales. That's not a bad thing. It's an easier story to tell. We had the opportunity. We had a colleague contact us and say, "Oh my gosh, you do this stuff. Maybe we could apply it to some whales." And obviously with an interest in conservation, and in particular, we worked on 2 different species. We worked on the bowhead whale, which the reason is because they're an Arctic species, and you may know there are still legal hunts among the native peoples in the north for bowheads, and so we were able to get some tissue from those whales that were legally collected. Otherwise, it's utterly impossible. We also worked on northern right whales, one of the most critically endangered species on the planet. The latest estimate I saw said there may be only about 400 left. You can't get tissue in any way. What we tried to do was get tissue from stranded whales that had died for one reason or another. But for the techniques that I've told you about, the tissue has to be fresh. If it degrades at all, we can't do the work that we do. So all of that was fruitless. Anyway, we were able to get some DNA from northern right whales and tissue from bowhead whales, and that's how we developed the story. So one thing I want to tell you before we get to the whale is about the food that they eat. The northern right whale loves to eat these little copepods, and these little copepods have a little red spot in the back. And again, we've got a spectrum here, and this is just the transmission profile instead of absorption. So if you shine a light through this guy, it looks red because it's got a red spot. This is the fancy way scientists tell you it looks red, all right? They look red. This is what they would look like to us. A cloud of copepods out there in the ocean. Beautiful. I mean, Who wouldn't want to eat that, right? Looks yummy. All right, so let me remind you, we see that beautiful blue and that beautiful red and any other colors that might be there because of this collection of 3 different types of cone cells. We all know and understand that probably more than we ever wanted to. But whales generally are very interesting. They are rare, extraordinarily rare among vertebrate animals, particularly among mammals. In fact, I think they are the only mammal that is known to have only one type of cone. They have rods, and they have a cone. Maximally sensitive in the blue, makes sense, they live in a blue world, right? But only one. So does a whale have color vision? Probably not. Unless somehow these rods and cones are talking to the brain at the same time. Probably. But, at least you've got a highlight cell and a low light cell, right? So if it's dim, nighttime or, late in the day or whatever, or they dive very deep, you can still see with your rods. If you're up near the surface, daytime, you can see with your cones. So life's still good. Maybe it's not very colorful, but it works, right? Poor old northern right whale, though. This is the sad part. Remember our opsin protein? The molecule inside these cells that actually detects the light, vitamin A plus this protein. We took some tissue from the northern right whale, and we sequenced it. So I don't know if you ever see these gene sequences. This is what they look like. There's a variety of different creatures over here that we're comparing, and this is just the sequence of the DNA of opsin, cone opsin, this particular cone opsin. We can find this cone opsin in people, we can find it in cows, we can find it in whatever, and compare it to the whale. And what's wrong here? What's wrong here? There is something very wrong here. In the northern right whale and some of its relatives, there is a mutation in that cone opsin that essentially chops the molecule in half. We've been able to look at the tissue from the bowhead whales. They still have the cell, but it is dead. It's functionless. So these whales, these big, beautiful eyes, they have rods, they have cones, their cones are dead. They have no color vision, absolutely no chance whatsoever, and probably can't see very well in bright light, if at all. And those masses of copepods tend to be near the surface. So we worry about, how do they feed at all? Maybe they can feed better at depth, or maybe somehow their rods are tweaked a little so that they can see a little better in bright light. We've actually done some work on the wiring diagram of the retina that I won't go into tonight, but we think they can still see, but we don't think they see well, and we think this may contribute to their decline, or at least exacerbate all the problems that face them. So, take home. Tarpon. Retinas change over the course of life in very dramatic ways. Changing color vision means that they have a retina that's built for best function in different habitats, new habitats. In species related to tarpon, we see very different changes in vision over the course of life. I only told you about 2 species. The tarpon and ladyfish are very similar. Eel is very different. Bonefish is also very different because it lives in a very different visual world. And finally, the tarpon has what we call pentachromatic vision, 5 different cone cell types. Amazing, really, really, truly at the extreme of mammals. Whales, these baleen whales, including the northern right whale, are at the other end of the spectrum. Whales are monochromats to start with, and right whales and their close relatives are the first coneless mammals that we know of on Earth. And I say coneless, functional cone. They have no functional cones. All right, so why should we care? Obviously, for the whales, conservation is a big issue. If we can understand their vision, we can understand what they see, maybe we can build legislation, build mechanisms to help protect them. One of the things we think about is longline fishing. You see these northern right whales very often, especially mothers, caught in long lines, and they are lethal. Now that we know the spectrum that they can see, we're hoping to inform the fishing industry, and maybe they could use different colored long lines that form a nice contrast to the blue world around them so that the whales can actually see them. Hopeful. And on the other hand, obviously, understanding their vision aids in conservation, aids in, making sure that pollution, eutrophication don't change waters in such a way that their vision is compromised. But one of the things I was about to allude to earlier that I didn't tell you before, but I should, is fish are amazing. And tarpon is a great example of this. Fish have nervous systems that can change over the course of life. They can fix themselves after problems. You always remember the old commercial, this is your brain on drugs. Or you hear, if you lose, hearing, go to that loud concert, you don't ever get it back. We have some capacity to repair nervous systems, but not much. Fish are amazing. They can radically change. They can radically fix problems over the course of life. Tarpon, when they go from a microscopic eye to a massive eye, there's huge amounts of change, and we've documented a lot of that change. What we and others have become very interested in is using the fish retina as a model to understand how the retina is capable and how the brain itself, the whole nervous system, is capable of modification over the course of life. And if you can figure out how they manage to do that, maybe you could use some of the mechanisms to tweak our own nervous system and fix problems from Parkinson's disease to blindness to who knows what else. So fish are a great model system for medical work. I don't bore you with all these names, but some of them you may have heard of or heard from over the course of time. I've had some amazing students in my lab, some wonderful collaborators from here in Florida, from Cornell, from Miami, from a lot of different places, and some fantastic support from regional, national, and world organizations. Couldn't be more thankful. Lots and lots of help to be able to collect data like this. So thank you. I'm sorry if I went too long, but thank you.
Anybody has any questions for Dr. Grace?
Questions, yes. So the question is, can the adult fish, the adult tarpon, continue to make rods and cones after it is mature? We haven't looked in the tarpon itself, but in other species of fish, it is known that they can. If you go in and damage the retina, for example, it can repair that problem. So we believe the answer is yes in tarpon, and we know it is in other fish. Yes?
Because like the whale and that, and probably
Mm-hmm.
would have the low vision.
Yeah, manatees, we've never done any work on, and honestly, I'm not sure if any has been done. But gosh, if anyone knows, correct me if I'm wrong. But in terms of whales, it is certainly possible, but the other thing you have to think about is distance. How far can they see? And I don't know if they would be able to visually detect an oncoming ship in time to move away from it. And then the other thing you have to think about is, and again, you'd have to think about the different species. Whales, I don't think they would, at least these whales, I don't think they would have any depth perception. Your depth perception comes from having eyes looking in the same direction, but laterally displaced from each other. So we've got two eyes side by side. And by comparing sort of the angle of that object out there, we understand how far away it is. But if you've got your eyes on the side of your head, you don't have any visual overlap. You don't have any depth perception. And I think that would be a problem for whales and manatees, actually. There's a lot of cool things that go on and things you have to think about. You always think, we do the same thing. Oh, this explains this. And you go, nope, wait. There's another problem to consider. So, yes. Well, yeah, so the question is, are we alluding to the idea that the whales are missing something in their diet? They're not able to get a particular food source or particular nutrients. we don't know, but obviously they have some kind of compensatory mechanism. They grow from calves to adults, so surely they're eating something. But but I think what it says, that was our first thought. Oh my gosh, these guys are blind. They're just. And we thought, oh, maybe this mutation just happened. But by comparing different species, we actually see that it exists in a variety of these different baleen whales, and even some toothed whales. So we think the mutation actually happened a long, long time ago. And so these animals somehow have been able to compensate. In fact, that's where we started looking at the wiring diagram of the retina. And again, I don't want to bore you or anything, but the short of it is, we think that over the course of time, over generations, many, many thousands of generations, the rods are sort of taking over some of the cone duties. So in other words, they're kind of feeding information to the brain through both the rod pathway and the cone pathway. However, they're still rods. So the whale can see, but it probably has very poor bright light vision. And we're trying to figure out, what do they really see? What does it look like to look out at the world through a whale's eyes? And how do they see those masses of copepods? Again, that's what they're filter feeders. And I guess when I was a kid, I used to think these big baleen whales just wander around with their big mouths open and take big gulps of water and squish it out through their baleen, and they capture a bunch of stuff. But they actually see these things. They see these clouds of copepods and go up and eat them purposefully. So again, we really don't know what effect this is having on them physiologically, or how easy it is for them to find food. They must eat, but my guess is it makes life more difficult for them in some way. I wish we knew better. Let me get one over there. So the question is, do the reflective crystals, that guanosine stuff, the reflective stuff in the tarpon's eye, does it interfere with daylight vision? I don't know. I'm really curious too. You would think it would. Again, if you think of your photoreceptors, sort of array of detectors, and this particular one is looking out at that particular point in space, this one is collecting light from that point in space. That melanin, again, keeps. If the photon comes to this photoreceptor and doesn't get absorbed by the photoreceptor, it goes through and gets absorbed by the melanin, so it doesn't go over and activate the wrong photoreceptor. And that's a good thing for bright light vision. The tapetum is doing exactly the opposite. So how this fish deals with that in the daytime, and they do feed in the daytime, I don't know what the answer is. I'm just as curious as you are, because it doesn't go away, and as best we can tell, it doesn't migrate. It doesn't move daytime. That's kind of what I expected to see, was it was going to move around. In fact, in many species, those melanin granules, even early on in these fish's lives, it's not just the photoreceptors moving. Those little pigment granules actually move down between the photoreceptors to shield them during the daytime, and they move back at night. But that tapetum, those guanosine crystals, as best we can tell, they're there day and night. We don't know what's going on. Keeps us employed, right? Yes, sir. Boy, if I tried to tell you right now, I haven't looked at that literature in a while, I might tell you wrong. I'm unaware of any others that have 5, but there are some that have 4. There are, bright light environment, lots of color, but I couldn't tell you which ones are which. But yeah, they do have, fish tend to do pretty well in color vision category. Honestly, we were so surprised at the tarpon, again, because, they're not living in a, shallow, colorful reef. They're out there in, near shore waters, but still. And I didn't point this out, I didn't point out that their most extreme photoreceptor is in what we call the ultraviolet. When we call ultraviolet infrared, we call them that because we can't see them. So our visible spectrum is from about, we say, 400 to 700 nanometers from blue to green, from blue to red. That's what we see. So if people tell you about the visible spectrum, that's the human visible spectrum. Lots of animals can actually see a little further into the infrared, a little further into the UV. So we weren't terribly surprised that the tarpon had one that's in a region we can't see. But what's really cool is most of those photoreceptors are all down there, the cones are down there in the blue part of the spectrum. So what we really think is they have this incredible ability to discriminate blues, which makes good sense if they're living in a blue world as opposed to on a coral reef. But yeah, there's a wide variety of different numbers and types in different species, yeah. Mm-hmm. I cannot answer that question. No. The question is what lure do you use? We were actually talking just beforehand. I think that there is a huge industrial application here. I've thought for a long time that my next life should be in the scientific lure design business.
If any of you do any tarpon fishing, you know that purple is an incredibly good color. And I think tonight we realize why those purple flies and purple lures might be so attractive to tarpon.
Gets those photoreceptors on the blue end going. So, yeah, but it's actually a great question in a scientific sense too, because if you're talking about lure design, if you're talking about lure use, you're talking about fooling the animal, which is what fishing is all about, right? And it's all about what they see. And, different species see differently. And in a tarpon, as an example, you've got obviously different stages of development with different forms of vision at those different stages of development. You've got very different light environments and very different retinal function in the day and the night. So there's a lot of interesting stuff there from a scientific perspective and from a fishing perspective. So again, somebody ought to be out there designing, making a living doing this kind of work and applying it to lures. Scientists don't get rich, trust me. You hear people sometimes talking about, the scientists just want to keep this argument going so they can get their giant grants and all that. Well, the grants pay for those students, and they pay for that microspectrophotometer, and they pay for a trip to the wastelands of northern Canada to get a little bit of eye tissue. Scientists don't make a lot, but if I were making lures, I'd probably get rich. Yes, sir. Mm-hmm.
I know their eyes. I wonder what
No. Do we know much of anything about sperm whale vision? The answer is no. Sperm whale, if I'm remembering correctly, the data says that they do have functional cones. But again, very deep, you wouldn't expect, if those cones work, if they work like ours do, you probably wouldn't expect them to be functional very deep. So, and again, that's the other thing. As a visual neuroscientist, I always think about things from a visual perspective, and I have to remind myself, I really have to remind my students, especially new ones coming into the lab, we sort of got our blinders on, so to speak, when we look at the visual system, but the animal, like us, they're using everything. And, they're like us. We don't think about it. We don't think about using our vision unless we're sitting in a room talking about it. But at the same time we're doing that, we're hearing, we're feeling the seat on us, we smell, we feel pressure, pain, temperature, all of these different things all at once. And all animals take in all of that information at the same time to make decisions about how to act. And so in terms of these, like the sperm whale or any other species, they're going to use what's available to them in the environment to inform decision making. So, they're probably, echolocating or sonar, to locate those squid as well, at least schools of animals. What light is available probably does activate their rod cells as well, but not their cones. So no color vision down there. There's no color down there anyway, really. It's pretty monochrome. What was I thinking of? This is all about fish and whales, but as you heard in the very beginning, I've done a lot of work on pit vipers and pythons, and they have this ability to literally, I'm going to say literally in quotes, see heat. They have eyes, they can see just like we do. but they have these other sensors, and they can see heat. And so we always have to remind ourselves that, they're really fascinating. You know, they can go in all these, like the sperm whale diving deep. The snake can be crawling around out here, and it can see every one of us with its eyes and with its heat sensors. If these lights go off, vision's gone, but the heat sensor's still working. They're perfectly fine. So think of that next time you walk outside at night and take out the trash. Yes, sir. We've done a little bit of work on them, and other people have done a little bit of work, but not much. A few years ago, I counted up about 20 papers on sea turtles. There's not nearly enough to really, understand exactly how they work and how the different species work in different stages of life. So, they're built like us. They have rods and cones. If I remember correctly, again, they have 2 different types of cones, so there's some color vision. But the thing that fascinates me is the kind of work that I've described here has not been done in them. A little bit, but not enough. And about the beachfront lighting legislation, I really wish we could get in there and do this kind of work, understand, so that you could really design the most effective beachfront lighting legislation. You know, the kinds of intensities and colors that we use today, they're based on extremely small amounts of science. It could be a lot better. You could have a wider range of lights for us to use and still not interfere with the turtles. You could make sure that turtles aren't interfered with, but we can still do what we want to do. There's not a lot, sadly. Mm-hmm.
Have you made any studies about color, correcting color vision?
Mm-hmm.
I don't know what the research is on. It was not feasible for him, but
The short answer is no. The question is, some people are treated with different colored lenses to enhance color vision, and I really have not looked into it at all. I've heard about it. I've been curious, but I've just never really. I really don't understand what it's doing, to tell you the truth. But it's a great question. I'm going to go home tonight and look it up because I really am curious, but I really have no idea how it works. And these are applied to developed individuals. This isn't during development. Yeah, and I don't know how it works or why it works. Yes. You mean changing color and pattern. This is a fascinating question for more than one reason. One is just plain fascinating. The other is we've been working on it. The question is, in species that can change color and pattern to match their surroundings, you mentioned flounder. Octopus are well known to do this. Cuttlefish, well known to do this. We actually, and I talked to my co-author the other day and said, can I mention this if it comes up? So, I didn't ask you to ask this, right? So nobody has ever known how animals are able to change their color and match their backgrounds. And we sort of know how they change color, but the second half of that's what's really fascinating. How do you match your background? How do you know what the background looks like? How do you know what you look like? We actually looked, and honestly, I think we were kind of fortunate in our choice because it forced us into a situation where we could explain even more. An octopus is all squishy, and it can look at its body and it can hold an arm up and all that. We decided to ask this question to try to figure this out in hogfish. Does anybody know hogfish? Really cool species, live around here. They're kind of stocky and they got these long, spiky bits on their dorsal fin, so you'd know them if you saw them. Hogfish can change color and pattern pretty dramatically. Hogfish cannot turn and look at themselves. They're a little too stiff for that. Their eyes look out laterally. They don't know what they look like, right? Well, but they do. And I'll tell you how. So, and you got to all be sworn to secrecy because we haven't published this yet. Literally, this is brand new. And it is so cool. We discovered, and this is work done very recently, it's with one of my graduate students who did some of this tarpon work. She's amazing, by the way. She's still here in Florida doing great, great work. But she's an avid fisher person. What do you call I don't know, fisher, okay. Yeah, an angler, there you go. So Lori always loved fishing from a boat. She spearfishes, she does, just loves anything in the ocean. And, we were just talking about the hawkfish one day, and she was like, man, they really change in dramatic ways. We should look into this. I said, well, let's do it. Well, so we applied some of the same techniques that we use that I presented here tonight. And what we discovered, of course, they have eyes. They can look around at the world, and they have color vision. They can see what's going on around them, but they can't tell what they look like. And so first of all, how do they change color? They change color because they have cells in their skin known as chromatophores, color-changing cells. These cells, some of them have the same melanin pigment we have. Some of them have a red pigment, some have yellow, some have other colors. And the way the cell works is there's all these little tiny pigment granules in these skin cells, tons of them in each cell. When those pigment granules are spread out in the cell looks that color, right? You got pigment granules all over the place. But there's these little railroad tracks in the cell that can gather them all up and do it pretty quickly. And when they gather them all up into a little spot in the center of the cell looks clear. So they spread out, cell's red. Bring them back together, cell's clear. Or another one, spread out, cell's yellow. Bring them back together, the cell's clear. So that's how they change color. And the brain, the hormonal environment, really the brain is doing this because it's fast, can send signals to those cells and tell them to get color or get clear. And you can do it with one type or two types or three types, so you can change the color in all sorts of different ways. And where you do it generates pattern, right? So you got these skin cells, change your color, change your pattern. You got eyes that can look around and see what the world looks like. But how do you know what the skin looks like? How do you know what do I look like now? And what am I going to change into? How do I know how to match this? It turns out, and this is crazy, they have photoreceptors. Like these rods and cones, they don't look anything like them, but they're like them in the sense that they detect light. All over the skin, thousands of them, and they are right underneath those chromatophores. So it's almost like each one of these little light detectors, each one of these things is like a little eye in the skin looking out at the world through color-changing sunglasses. So they got eyes to look around at the world and see what it looks like. Then they got all these light sensors in their skin to look through their own skin and know what they look like and know what they're changing into. It is the coolest thing ever. So,
Well, unfortunately, guys, I love when these question and answer sessions go on for a while, but the library does need us out here in the next couple minutes. Can we get another big thank you for Michael?